This lecture is going to be on Chapter 3, Red Blood Cell Production, Function, and Relevant Red Cell Morphology. At the end of this lecture, the students will be able to outline erythropoietic production from origin to maturation, with emphasis on stages of red blood cell development, describe immature red blood cells with regards to nucleus, to cytoplasm ratio, cytoplasm color, nuclear structure, and size, clarify the role of erythropoietin in health and disease, identify the three major red blood cell metabolism pathways essential for red blood cell survival, describe the composition of the red blood cell membrane with regard to key proteins and lipids, differentiate between microcyte and macrocyte and indicate the conditions in which size variations are seen, indicate the clinical conditions in which variations in hemoglobin content are seen, describe the clinical conditions that show polychromatophilic cells, identify the pathophysiology and the clinical conditions that may lead to target cells, spherocytes, ovalocytes, elliptocytes, sickle cells, and fragmented cells. List the common red blood cell inclusions and the disease states in which they are observed, and describe the value of the reticulocyte count relative to the bone marrow function. When it comes to red cell production, it all starts with the pluripotent stem cell. So you kind of think of that as the mother cell. And these cells can be very versatile in all the different cells that they can produce, but right now we're just going to be talking about red cell production. The erythropoietin, or the EPO, is produced by the kidney, and this is a hormone that stimulates the red blood cell regeneration, so it plays a vital role in red blood cell production. Once the red blood cell meets full maturation, the approximate lifespan is 120 days. There are several changes that occur during the red blood cell maturation. The cell size reduces, the nuclear to cytoplasm ratio decreases, the nuclear chromatin becomes more and more condensed, the color of the cytoplasm changes, and it becomes anucleate, which means there's no longer a nucleus within the cell. Here's a diagram that shows the different stages of the red blood cell, and as you can see, we start with the pronormoblast here, and you can see that the nucleus to cytoplasm ratio is primarily nucleus with a much smaller area of cytoplasm around here. Then we move into from the pronormoblast to the normoblast. You can see the size of the cell overall has become smaller. You're seeing more of the cytoplasm. You can also see that the chromatin pattern here in the nucleus is becoming more condensed. Then we move on to the polychromatophilic normoblast, much smaller. The cytoplasm is much more apparent. The ratio has shifted. And as we move into the early orthochromatic normoblast, you can see it's even smaller. That nucleus is super condensed. It looks like a solid color. You really don't see any striations within the nucleus of the chromatin. Lastly, the last stage we have here with a nucleus is the orthochromatic normoblast. You can see that nucleus has shifted. It's a kind of eccentric instead of being more in the middle of the cytoplasm. The next stage is the reticulocyte. Can you see there is no longer a nucleus, however it still has more of a purple color due to the RNA still being within the cytoplasm. And lastly, we have a normal red blood cell. It's going to have more of the pinkish salmon color, and that's what you would anticipate seeing on a normal CBC differential. So here we're going to go through each stage that we saw consolidated on the slide previously for the red blood cell maturation process. So we start with the pronormoblast. Here are the criteria that you look at for cells, not just for red blood cells, but also for white blood cells, as we've talked about. So we look at the size, and for the pronormoblast, that's 18 to 20 micrometers. The shape of the nucleus is round. As you can see, we have an arrow pointing to one in particular. Nucleoli may be visible, and you may see 1 to 2 nucleoli. The nucleus to cytoplasm ratio is 6 to 1. You can see that primarily, as we just discussed, the cell is primarily a nucleus with less cytoplasm on the periphery of that nucleus. The chromatin pattern is fine. The cytoplasm stains with a dark blue or purple color, kind of a, I think they call that cornflower blue. And here at the bottom, here's your reference as to check what's normal, what's normal. What should you see in the bone marrow, which is the BM here. That's 1% of your bone marrow, you would have pronormoblasts. And in the peripheral blood, your circulating blood, the blood that you will be testing when you do a complete blood count in hematology department or making a differential smear, you should see 0 of these there. So if you were to see them, you'd know that something is happening with the patient and needs further investigation. The next mature state of the red blood cell is the basophilic normoblast. Here you can see the size is 16 micrometers, so it's a little bit smaller than the pronormoblast. The nucleus is also round. Instead of seeing 1 to 2 nucleoli, you may see no nucleoli or may up to 1 nucleoli. The nucleus to cytoplasm ratio is the same. The chromatin pattern you can see is much more condensed. You can see it's clumped up more. You see little white spaces in between. The cytoplasm is cornflower or a darker blue. And again, you would see these in the bone marrow 1 to 4%, and you should not see these in the peripheral blood as they are. The immature stages should only remain in the blood marrow unless there's some sort of disease state happening with the patient. Next, we have the polychromatophilic normoblast, and you can see just in this image here, it's next to appears to be the basophilic normoblast. Here is the basophilic normoblast we just talked about in the previous slide. Here you can see the arrow pointing to these two cells, which is the polychromatic normoblast. You can see it's much smaller. The nucleus is still round. You should see no nucleoli. The nucleus to cytoplasm ratio has shifted. You have more apparent cytoplasm than you have in the previous two stages. The chromatin pattern is going to become much more condensed. You can see it looks more consolidated, especially compared to this cell. The cytoplasm is blue layered, meaning you can see various colors within that cytoplasm. In the bone marrow, the polychromatophilic normoblast should be 10 to 20% of what is found in the bone marrow, and it's still 0% of what you should see in a peripheral smear of the peripheral blood. Next, we have the orthochromic normoblast, otherwise known as a nucleated red blood cell, which is here. And you can see that, again, the cell itself overall has become smaller, 8 to 10 micrometers. The nucleus to cytoplasm ratio is 0.5 to 1 or a 1 to 1 ratio. The nucleus is still round. We should have no nucleoli, and the chromatin pattern has become fully condensed, so it looks like a solid nucleus. You really don't see any strands or any type of chromatin pattern. The cytoplasm is increased, which, obviously with at least the 1 to 1 ratio. You can have kind of pink, purple, different undertones almost layered in this cytoplasm. This is the last stage before we lose that nucleus, and it looks like a fully mature red blood cell. Again, in the bone marrow, these are going to be 5 to 10%. In the peripheral blood, what you see in the hematology department, these are typically not normal unless you're dealing with a newborn or the patient has some sort of disease state that is triggering these to be released into the peripheral blood. The next stage is the polychromatic macrocyte, otherwise known as a reticulocyte. Now, this is not the same stain we've seen on the previous slides, which is the right stain, which is what you will be using primarily in the hematology department of the laboratory. This is a supravital stain known as new methylene blue. And what it does is it shows the remnant of RNA that's left over from the red blood cell working through the maturation process, and it points to that is a new or newer red blood cell that's been in production and has been released from the bone marrow into the peripheral blood. And what you can see here is the remnant of that RNA within the cell, and it's visualized as the reticulum. So you can see here, obviously we've lost our nucleus, and typically reticulocytes make up 1% of what's happening in the bone marrow, and in the peripheral blood you can have 0.5 to 2%. Here you can see a side-by-side of a reticulocyte. On the left side, this is the right stain that I mentioned previously, and you can see that these cells have more of a purplish hue under that right stain and less of the pink color. And under that new methylene blue, you can see the remnant RNA or reticulum, which is the darker color within these cells here. And lastly, we have the mature red blood cells. So we've lost that nucleus. We have the correct color under the right stain. The average size or normal average size for a mature red blood cell is going to be 6 to 8 micrometers. You'll see that the tool that we use in Cellovision shows you, you can slide that over the red blood cell to see is this 6, and if it's a little bit larger but it's still normal, it can be 8 micrometers. So the appearance of the mature red blood cell is a disc shape, and it has an area of central pallor. Some of these you can see better than the other. This one you can see a central pallor. This one here where it looks a little bit lighter in color. Here's one here. Almost like a donut disc type shape. Here's a consolidated chart that shows the criteria of a mature cell versus an immature cell in regard to the red blood cells. So typically an immature cell is going to be larger, which is greater than the 8 micrometers. As we just discussed, a normal red blood cell is going to be 6 to 8 micrometers. The color of the nucleus for an immature red blood cell is going to be more in the blue areas, the blue hues, whereas a mature red blood cell is going to have the salmon or pink colors. You're going to have a nucleus with the immature stages, but the mature stage has lost its nucleus completely. The nucleus to cytoplasm ratio is going to be much higher in an immature cell and very low when it comes to a mature cell. So the red blood cell membrane is designed to aid the body in carrying your hemoglobin. It delivers it to all over the body as needed. The structure is trilaminar, meaning it has 3 different layers. The outer layer is consistent of glycolipids and glycoproteins. The central structure is made up of cholesterol and phospholipids, and the inner portion is the cytoskeleton, and it has very specific membrane proteins there. It has integral and peripheral proteins. The overall structure is made up of 50% protein, 40% lipid, and 10% cholesterol. The integral proteins are active in the transport, active and passive transport, meaning it actively transports things but also stands by ready to transport. The structure for some of the antigens are the glycophorin A, B, and C, which is the point of where the antigen is able to attach. The peripheral proteins, which make up the cytoskeleton. The cytoskeleton is responsible for the deformability, meaning the red blood cell is able to shift its shape or bend to conform to move in and out of very tiny vessels. And it also, the cytoskeleton also provides the elasticity needed for it to do this as well. And the cytoskeleton is made up of spectrum and anakyrin. The transport mechanisms are used to diffuse freely throughout these special channels, and they want to transport things such as water, chloride, and bicarbonate. It's more highly regulated with an intracellular extracellular ratio when it comes to your metabolites or electrolytes such as sodium, potassium, and calcium. Since a mature red blood cell has no nucleus or any mitochondria, it is dependent on getting all of its energy from the breakdown of glucose. So within that red cell metabolism, there are 3 ways that it can acquire that. The first way is the Embden-Meyerhof pathway. That provides 90% of the ATP or energy that it's needed. The second way is the phosphogluconate pathway, which provides 5 to 10% of the ATP. And lastly, there's the methemoglobin reductase pathway. And this keeps the iron in a reduced ferrous state so that the hemoglobin can bind oxygen to it and then therefore deliver it to the tissues of the body. When it comes to our job as an MLT in the hematology department of the laboratory, we rely strongly on automation, and we'll learn more about that, or we have learned about that in Chapter 1. But those instruments are set up to flag discrepancies or abnormal features within the blood. One of the ways that we can verify that is by preparing a quality peripheral smear, which is used with a drop of blood and pushing that along the slide, adhering that to the glass slide, and then staining it with the right stain. When we review that slide, we're going to look at least 10 fields where the red blood cells are not touching. You have some sort of gap. Then you examine the morphology of those red blood cells. Are you consistently seeing that morphology in every field? Can you determine whether or not that morphology is artificial, meaning it has been created by an irregular push of the slide, something that happened during the process of making the slide? Or is it actually pathologic or pointing to some sort of disease state? So some of the abnormalities that we look at are variation in size, which is known as anisocytosis. We also look at a variation in shape, which is known as poikilocytosis. When it comes to evaluating the anisocytosis or determining whether or not there is abnormal cell size, you first have to be mindful of what normal cell size is, which we just discussed, is 6 to 8 micrometers. Your MCV should be within the ranges of 80 to 100 femtoliters, and your MCHC should be 32 to 36%. Then so those are the parameters for a normal red blood cell. When a red blood cell is considered macrocytic, meaning it is larger than the normal size, then the size is going to be greater than 9 micrometers. And microcytic would mean that it's smaller than normal, and the size would be less than 6 micrometers. When it comes to reasons or disease states that you may see microcytic or smaller than normal red blood cells, one instance is with anemia, specifically with iron deficient anemia, where you have an impaired iron metabolism that causes that issue, inflammation tied to anemia, thalassemia, where you have diminished alpha and beta chains of the red blood cell molecule, sideroblastic anemia, and even iron overload disorders such as hereditary hemochromatosis. When it comes to seeing macrocytes or macrocytic peripheral smears, some instances that can cause the red blood cells to be larger than normal are a vitamin B12 deficiency, a folic acid deficiency. Liver disease can affect this, as well as any type of megaloblastic anemia. And typically with the red blood cells being larger, your MCV with megaloblastic anemia is going to be greater than 110 femtoliters. You can also have variations in the red blood cell color. You can have polychromasia, which you can see here in this slide. The arrow is pointing to a cell that looks much more purple than the other normal red blood cells. This is because, as we discussed previously, with a reticulocyte, there are remnants of the RNA in there, and it looks purple with the right stain, and it's going to look blue with the new methylene blue stain, which is what we saw an image of earlier. The cell size is going to be 10 to 12 micrometers, which is larger than the average mature red blood cell. And this is happening because there's a bone marrow response due to the anemic stress that the body is under. Hypochromia indicates that there is a lack of color or a lack of hemoglobin. You're going to have a much larger central pallor than what you see typically in a normal red blood cell. You can see that I've drawn some red lines pointing to ones that are particularly very hypochromic. The hemoglobin synthesis has been impaired, therefore the amount of hemoglobin is decreased in the red blood cell. You're going to see an MCHC that's less than 32%, and typically this is seen in iron deficient anemia, thalassemias, and sideroblastic anemia. When it comes to shape and color, we have the spherocyte indicating that it is perfectly round, which most red blood cells are not. They have a little bit, they do have a round shape, but not perfectly round, and they're not as compact, and they have a very concentrated pink color where you do not see the central pallor. You can see the arrow here pointing to a spherocyte. It can be connected to hereditary spherocytosis, which leads to the spectrum abnormality. The spectrum is part of the cytoskeleton structure of the red blood cell. It can also be tied to autoimmune disease where the antibody-coated cells are affected. You're going to see an increase in the MCHC greater than 36% and osmotically fragile, meaning it has a difficulty in maintaining what's outside the cell and inside the cell because it has lost its flexibility. These are sickle cells, and they are a half moon shape, and they typically come to a point on one or both ends. You have reversible and irreversible, where the irreversible are more crescent shaped and have those pointed projections. This is due to the sickle hemoglobin or hemoglobin S. It forms tactoids under hypoxic stress and then therefore causes hypoxia. When it comes to poikilocytosis, as well as the sickle cells, we have ovalocytes and elliptocytes. Ovalocytes are going to be more egg-shaped. Those are typically associated with thalassemia or megaloblastic anemia. And elliptocytes, which are more elongated or cigar-shaped, occur due to an abnormal spectrin and protein 4.1. It can be tied to iron deficient anemia, hereditary elliptocytosis, and idiopathic, meaning with no specific disease state, myelofibrosis. Target cells can also be seen. They have the standard bullseye shaped. You can see here the two arrows pointing to those. Sometimes these can be due to artifact, meaning the way the push of the slide occurred. It can make a wonky looking cell, and it may not necessarily be a target cell, so then you have to evaluate the wet work of the CBC. Does this line up with any abnormalities, as well as the quality of the slide. How many of these cells are you seeing? Are you seeing it consistently in 10 different areas of the slide when you move around on there, comparing what you see from field to field? They are due to a decrease in the hemoglobin, but they also have an increased red cell surface membrane. So there's not enough hemoglobin there to fill up the red blood cell. So this can be seen in iron deficient anemia, hemoglobin C disease, liver disease, and post-splenectomy, meaning the spleen has been removed. Stomatocytes can also be seen. They have an elongated area of the central pallor. You can see the red arrow pointing to the stomatocyte there. It can be induced by chemical agents, or it can also, as I referred to with the target cells, be due to poor peripheral smear slide technique, meaning as the slide was pushed, it sort of ripped out the center of a few red blood cells. Again, you want to compare different fields. Are you seeing it consistently in each field, or is it just a few here and there, and you could chalk it up to just a bad slide? Stomatocytes can be seen in hereditary stomatocytosis, and they can also be seen in alcoholic conditions. Acanthocytes are smaller red cells that are surrounded by an uneven thorn-like projections or spicules. You can see the arrow pointing to the acanthocyte in this image. It's due to an excess of cholesterol. It can be seen in liver disease, autoimmune hemolytic anemia, and LCAT deficiency. Bite cells are a type of fragmented cell, and as the name indicates, it looks like there's been a bite taken out of the red blood cell. You can see the arrow pointing to one here. It's also known as a helmet cell because you can see the similarity of it looking like a helmet or looking like someone has taken a bite out of it. We also have other fragmented cells known as schistocytes, and I know this image is a little pale, but schistocytes are various shapes, so you can see in each image, A through I. They're not a normal red blood cell, that's obvious, but they aren't any particular shape. They're not necessarily hyperchromic, but the presence of them being there, especially in numerous fields, should be noted on the patient's results, excuse me. Here is a burr cell which has almost like a bumpy periphery. It's not spiky like the acanthocyte, it's more of rounded out. We also have abnormalities with red blood cell inclusions, and there are various names, and here's just a little chart. I'm broke it up a little bit with images in between. We have inclusions called Howell-Jolly bodies, and the composition of these inclusions are DNA remnants. They can be seen with patients who've had a splenectomy, or meaning their spleen has been removed. Hyposplenism, meaning the spleen is not working as it should be. Megaloblastic anemia, hemolytic anemia, and pernicious anemia, which is tied to the B12 and folic acid deficiency. We also have Pappenheimer bodies, which are composed of iron. This also can be seen in the splenectomy, and I also want to point out, as I mentioned previously, the spleen is a filtering system for the blood, so when the spleen has been removed, those abnormal inclusions or abnormal red blood cells are not clean or filtered out as they would be. So that's why you're going to see that listed as a potential reason for these things happening. So also with the Pappenheimer bodies, it could be tied to hemolytic anemia, sideroblastic anemia, megaloblastic anemia, and hemoglobinopathies. Here is a good example of what Howell-Jolly bodies look like. They look like one little tiny purple dot. And you see I've taken my red marker and pointed to them. So this is what that looks like. It's a DNA remnant that the spleen, or for whatever reason, those inclusions have not been cleaned out, or those red blood cells have not been moved from the peripheral blood. Here's a good image of the Pappenheimer bodies, which are composed of iron. You can see it's much smaller, and you usually see 2 to 3 little dots together. They're usually consolidated together is how they look in this particular image. We also have more red blood cell inclusions, such as basophilic stippling. The composition of those are RNA and mitochondrial remnants. Conditions that can cause this are lead intoxication, thalassemia, and abnormal heme synthesis. We also have Heinz bodies, which are denatured hemoglobin. The conditions can come from unstable hemoglobin, hemoglobin xuric, hemoglobinopathies, and G6PD deficiency. Here is an example of the basophilic stippling, and you can see where it's teeny tiny little dots. Some are larger than others throughout the red blood cell. It's a purplish pink color, not quite as purple as the Hal Jolly bodies, but they have more of a purple tint than a pink tint. As we talked about earlier with the new methylene bloom stain, this is an example of that, of showing you what Heinz bodies look like on the new methylene blue stain. We also talked about the reticulocyte, where we have remnant RNA that causes the red blood cell on the right stain to have more of a purplish hue, but when it comes to examining it on the retic stain, or the new methylene blue, you can see the tiny dots. It's the quantitative reticulocyte count is the most effective means of assessing red blood cell generation. It's an important indicator of how the bone marrow is functioning. A normal retic count for an adult is 0.5 to 2%. It's going to be much higher in a newborn because their body is spitting out those red blood cells, and it's a much higher count of different range for newborns. So that's going to be 2 to 6%. Reticulocytosis means that there's an increase in the reticulocyte count, and sometimes this is appropriate. It's the body's way to balance out any type of anemic stress, so it's an appropriate response to that. Reticulocytopenia means that there is a decrease in the reticulocyte count. This is usually coming from an ineffective erythropoiesis that occurs in the bone marrow, or any type of aplastic condition, meaning the processes that should be happening, regeneration, maturation in the bone marrow, are not occurring as they should.